missing one thing, but it's okay. All right. Can everybody see that?
Yes.
Yes.
Okay. All right. So this is I put together a presentation for that paper that I wrote on. I just had an idea and said, well, let's see what comes up. So I'll explain what that is. Okay. So from genes to tissues and from tissues to neurology. So I'm proposing a new roadmap for autism research and treatments. A little bit of motivation. Everybody knows that I've had a very hard time trying to advance a model of neurology for autism. Right now, the model is behavioral. So I decided that maybe one way to circumvent the whole behaviorism approach would be to adopt a popular framework in other fields of medicine and treat autism as a medical condition that we could personalize treatments and diagnosis and everything. And so there is this model of precision medicine, which has these layers of knowledge that are accumulated from the person. And for each patient, you get all the contributed data from surveys and questionnaires, and then you get clinical tests, and then you get an analysis of behaviors, which usually is by observation and description, by eyeballing the person. You have also some history of the exposures to environmental toxins or geographic, if the person lives in a highly polluted area or with pesticides and so on, that also contributes to the overall knowledge, the microbiome, and the genomics. Now, what happens is they combine all this information, and this has been an approach useful in cancer treatments for highly targeted cancer treatment. So instead of doing massive radiation or massive not radiation, that's pinpointed, chemotherapy or gross treatments that destroy everything and so on, what they do is they take this approach all the way to genomics and develop a treatment that is more in tune with a person's network, all these layers of the knowledge network for that particular individual. That's why there is a little red guy here in the middle of the crowd, because instead of a one-size-fits-all model, it's a personalized approach. So I decided that maybe for autism, this is something that would help us basically change the way in which is diagnosed and treated, which is right now a one-size-fits-all model. It gives rise to a very heterogeneous disorder, and then it's problematic. So that's what I just explained. So this is, instead of a one-size-fits-all medicine, you stratify patients' types by depending on criteria derived from all these layers, and then you do an individualized treatment design. But in so doing, I came upon this stumbling block, which is the behaviors is completely done in a observational manner, which is insufficient because it leaves out a lot of details that we want to know about the patient's biorhythms, for example. And so then came along the biosensor revolution, and our lab defined these movement classes in a phylogenetically orderly taxonomy of the nervous system, meaning there is an order of maturation that we have been finding matters in autism, and combining those, we decided to come up with a digital approach to the whole behavioral layer of this precision medicine platform. So we have developed this intelligent behavioral analysis, IBA, and it really is to complement the current dominating treatment intervention in autism called ABA, Applied Behavior Analysis. So we could then combine the kind of measurements that they take visually in applied behavior analysis with off-the-shelf technology that is affordable and people can have it at home, and it's wearable, and it's advanced in a way that we will soon, in a few years, all be able to walk around collecting a lot of data. We already do that, and it's going to be critical for the whole social distancing in terms of the continuing our behavioral research in behavioral neuroscience and in psychological social sciences and so on. So decided to do that, and then add that layer of digital behaviors, and then follow these proposed phylogenetically orderly taxonomy, which we've been using in the lab quite a bit to organize our protocols and our thoughts, and then probe the nervous system along these different layers of neuromotor control. Now, to these layers of neuromotor control, autonomic, involuntary, and voluntary, there corresponds three fundamental types of muscles, which we have in our nervous system, which produce all the motions that we end up performing. The autonomic level with the cardiac muscle that you find in the heart. There is the smooth muscle, which you have things like the bladder and in the intestines and so on. There, and that's involuntary, and it goes quite often after 60, 70 as the person ages, and the functioning, the involuntary motions start. We're talking micro motions here. It's very, and then we have the voluntary movements mediated by skeletal muscles contractions, and as we have learned in the lab, this has a layer of efferent information flow going from the brain to the body and to the peripheral nervous system, and then also a reafferent kinesthetic system flow of information feeding back to the system that controls to the controllers. So most of neuroscience is doing a top-down from the brain into action and so on approach, and we're closing the loop by doing a bottom-up approach, considering all these levels. So that's kind of the big picture. And again, the new roadmap that I'm proposing is introducing these digital behaviors, which add a new layer of granularity and combines, non-trivially augments the former behavioral analysis as we know it. The important thing here, I stress in the paper, is that by taking this approach, we can connect to genomics, because right now the big problem that we have is there is a gap here between behaviors and genomics, and there is some kind of circularity where you have observational clinical criteria guiding the basic science. And this is very odd, because often it's the other way around. It's science which guides the clinical approaches by at least a decade or two of knowledge, and then the clinical criteria updates according to the scientific advances. In autism, the scientific advances are a little bit stuck because we're backwards. Everything that is done, so if you take genomics, it's not blindly done at all. It's done based on clinical observation, and so you can't get out of that loop. So in this new stratifying approach, what I'm trying to do is take advantage of all the maps that already exist in the system from head to toe. So for example, the whole facial recognition and emotion and sentiment analysis and all that, we can base it on nerves that already have been mapped out in the face and that are involved in all the social axes for eye movement, hearing, swallowing, speech, and so on. And the rest of the body, we got the dorsal root ganglia nerves, and we have a very sophisticated map in the periphery that maps also to the somatic sensory motor areas. And we have very precise subdivisions of labor here between the central and the peripheral nervous system. And within the peripheral nervous system, we have several subdivisions in the somatic and the autonomic nervous system. And we also have the enteric division with the gastrointestinal functioning, the parasympathetic and the sympathetic division. So this allows to organize our research, our basic research, very nicely. And what I'm proposing is that we bring this to the clinical domain, which is primarily it's a very gross approach. It's primarily done by what we can see exclusively. So we can add a layer of the invisible, like whatever is hidden to the naked eye, we can get digitally profiled. In autism in particular, there are all these somatic sensory motor issues that exacerbate with age. And because there is this behavioral approach right now, what we have is pretty much, you know, all of that has sidelined the neurology of autism, but we have now accumulated a very large body of knowledge where we know that there are dysregulations in the nervous system across all these layers that I'm proposing that we that we examine and that we based our diagnosis and our treatments off. And so among them are the ataxias and the traumatic brain injury because of frequent falls, the fear of fall, which leads to depression and the person not moving too much and not leaving the house, which is, compounds with depression and so on. And we have a whole number of issues in the related to these cranial nerves, which also are important for the social axis. So in other words, because these neurological issues are not treated at all or addressed from an early age, they really accelerate and they the aging of the person with autism. And so you see that by 40 years of age, we have 20% of autistic adults showing Parkinsonism signs, whereas in the regular population, we only see a 0.9% in the after 65 or 70 years of age. And so we've explored large databases in Abide, and we've confirmed that indeed the adults have a much different profile, for example, in the involuntary movements than the controls, and decay much rapidly in an accelerated rate. So all of these, okay, I thought, okay, things don't add up in autism. There is some kind of agenda pushing for all of these behaviorism. They define as a behavioral disorder. There's a behavioral pipeline, which is a behavioral diagnosis and then a behavioral modification treatment. And it's all a burden to the person being diagnosed or treated. In the case of children, it said that they behave socially inappropriately, or in odd ways, that this needs reshaping, or based on punishment reward regimes, which are based off operant conditioning or animal operant conditioning that were translated to human children without any kind of uh, you know, consultation to the developmental um, neuroscience or the neurologists that study children, you know, child development, or any other members of the scientific community were consulted on this. This was done just in the 70s without, I mean, it was, to place things in perspective, there was a crisis, you know, there was nothing back then. So this was, in retrospective, is, you know, now it's easy to say and criticize, but at the time, one has to be fair that this was a solution that at the time was revolutionary. But, you know, it's been of 40 years of this kind of thing, and 2 decades of neuroscience research that we need to update these things. So instead of saying, you know, the person walks odd or has, or is uncoordinated, or has ritual and repetitive behaviors, or does not make eye contact and therefore must be antisocial, or must have no feelings, or must have no empathy and whatnot, instead of having this kind of negative deficit view of the person, we could do better and actually examine the neurology underlying that, underlying all those seemingly odd behaviors, which are all have a neurological explanation. And so here's on the right hand side the other alternative view. So in the 1970s, a little bit of history, there was a neurological model proposed by Ralph Moore, who passed away already, and Antonio Damasio, who is currently a prominent philosopher at USC. This model was competing with Lovaas' nascent model of behavioral conditioning for kids, which have been translated from animal models to like a Skinner conditioning to human kids. This was happening at UCLA, and in parallel, there was a model of genetics pointing at several possible candidate genes, and this was by Ridbo and also at UCLA. So these 2 people were competing there. That's where everything really originated, that was revolutionary at the time. This model was abandoned, this neurological model was pretty much, it was in the background, sort of a lot of other people were trying, but they just didn't win this battle. I mean, the behaviors got very well organized, they took over, and that essentially is what we have today, this behaviors model. So eventually we have this autism diagnosis observation schedule, guiding and informing and steering the genetics. And so that's what I'm talking about. There's this circularity in this process where you have, it's either give me a subjective ADOS account of the population and then do genetic testing on those people who already have a label, and then you have a score to that genetic condition. Or given a genetic condition you already know, do an ADOS, and if it has autistic traits, then you scored a genes high for autism. That's so the criteria, and it's the criteria I actually used in the SFARI genes module, which is the Simons Foundation Autism Research Initiative, and they have all 900 something genes linked to autism in this way. Either they had the genetic diagnosis and they had autistic traits, or they got an autistic autism diagnosis, and for whatever reason, either through research or maybe they have medical conditions that require genetic testing, or maybe the parents had a family, you know, autosomal dominant, or somebody else in the family got it, and then they decided to go do genetic testing, and then because they had a diagnosis of autism already, then that gene is counted, and the frequency then defines kind of the score, the frequency across the population and across the paper. So the SFARI genes module is amazing because it has all these compilation of papers and evidence and so on. And I thought, why don't I have a stab at it? And given that they have used criteria that deliberately sidelines any neurological stuff and claims it's behavioral, and I use that, and let's see if I can get something, anything. Let's see what happens. So at the moment, there's no blind way to do this, to say, okay, give me a random draw the population, and what are the odds that this is a genetic condition that also leads to autistic traits. We don't have a blind way to do it. So pre-label with this ADOS or DSM criteria, or give me autistic traits and deficits or differences in social interaction and communication repetitive disorders, and what likely would be the genetic condition. So there's nothing like that. Those are the current autistic traits. But what I want to do is augment that with a neurology that is underlying the whole model. So why not go back to the neurological model of autism that we had competing in the 1970s? And here's some argument for that. We've had 2 decades of brain science. In the US, these have been advanced by the Decade of the Brain Initiative, which was under Bush, under President Bush, and under the Brain Initiative, which was under President Obama, is still ongoing. All the conditions in autism have a neurological explanation. And there are other disciplines out there that provide accommodations for these neurological conditions. So in any case, you know, so they help the person improve quality of life. These are, for example, the ataxia, Parkinson's disease, mitochondrial disorders, and so on, which if you go into the genetics, you see that a lot of the phenotyping they describe is present in autism. So why don't we go backwards and find neurological conditions that have genes that may also go on to receive a diagnosis of autism, and begin to characterize those individuals based on the knowledge that we already have of the neurology. Because the worst case scenario is that, there's no worst case scenario, because you actually take advantage of what another field already is doing, and you might be able to leverage some of the targeted treatments that they have, which they do. So I reason that these conditions all have these known genetic origins that are well known as neurological conditions. So what if those genes are also present in autism, as defined by these ADOS and DSM criteria, which precisely and specifically sidelines neurological issues? So that's like the worst, you know, case scenario. Like I'm taking the criteria that precisely says this is not neurological, and let's see if we come up with what we get. So find the ataxia, Parkinson's disease, X chromosome genes in the literature and remove them from the full human genome and see what we get. And remove the autism genes as determined by the SFARI criteria, using exclusively the ADOS and the DSM criteria, sidelining the neurological conditions, see what we get. And then find the genes in the SFARI gene modules common to these neurological conditions, and remove the rest of the genes and see what we get. So in other words, so that if we find something that is neurologically like, it's not because of those conditions. So I removed also the genes that were overlapping, the intersection. And so even if we remove the intersection with those genes, what do we get? So do we get gene expression in the 54 tissues pointing to neurological issues like motor, sensory, emotion, memory? So this is what I use. I use the SFARI genes module. And if you click on I'm going to upload this presentation. If you click on this, you will go to the SFARI genes. I'm not going to click on it for the best interest of time. And this is the GTEx portal, which is also where all those genes, human mRNA information is, you have everything that you need there for the full human genome, and it's being updated constantly. And another thing that I used was this online Mendelian inheritance in man database, which you type in the gene and you get the whole shaban on all the phenotypic description, treatments, existing support systems, and whatnot. These are all open access, by the way. So this was just something I did all by wondering, what if, I didn't know anything about any of this before I started this project, kind of on autopilot kind of thing mode. I just started wondering, what if this, what if that, and then what exists, what do we call, what can we find about X or Y, and then found all these sources. And so they are open. And again, the literature, I went into the literature and found genes that have been very well characterized and have these neurological labels. So neurology it's also like behaviorism, it's by observation, but because it relies a lot on movement and motion and sensory, it has a better, a more refined kind of criteria to support and treat the person. So the main message here is that if you're doing basic science or clinical, translational science to clinical practices that is not supporting the person, it's no good. I mean, in the ataxias, it's so good because you go to an ataxia conference and the patients are the ones telling the researchers what they need. I need you to do research on how to stop or slow down the progression of my muscle issues, because I need to walk 5 more years. I want to use my hands 5 more years. So please slow it down. So the researchers are, doing that kind of research to help the person. In autism, nothing like that. It's all deficit. Oh, this person, this kid is odd, is antisocial. There's nothing here to help them. So that's the whole motivation. So here I listed in the table the number of genes that I used. And as you can see in the autism cases, the largest, 906, have been documented. And these numbers here are by the score. So 144 are the first score, and score number 1, 260 number 2, and so on. And 78 is syndromic, which is more complex. The scoring criteria I describe in the paper, and it has to do with a combination of literature, support, number of references, and direct implications, familial ties, and so on. You can read in the paper about that. I just took it straight out of the SFARI site and the literature. Okay, so that's sort of what I did. And then I took the data from the repositories, which consist of tables. These are matrices of RNA sequence genes. They have 56,146 of those along the rows. And then they have 54 genes, tissues, sorry, like the amygdala and all the tissues that they look at gene expression, these gene expressions. Those are on the columns. I transpose that matrix and then express each of the tissues as a function of the 56 to 146 genes. And then for each one of those, I removed the genes selectively for each neurological condition to see what happens, which are the genes, which are the tissues that are most affected when you remove those genes. So it's a very gross kind of exercise here, but it's a stochastic process. So I said, okay, so this is a random kind of reorganization of the whole gene expression. Let's see what happens. So I express each tissue as a function of the gene expression, which is denoted by this transcript per million, TPM. I took the log of that because these are very large numbers, and then you can better visualize everything with the log scale. And each individual count I normalize using this normalization which considers the entire average of the global count for all the genes and all the tissues, and the maximum for each tissue. So here's an example of what you get in the GTEx. These are called violin plots, and in the middle there is like the median value. In the horizontal on the horizontal axis you have in alphabetical order all the 54 tissues that, you know, so adipose, you can read it, you know, but so for example, if you type in Shank3, which is a condition that we have studied in the lab quite well, and they have very odd, you know, gait and tremendous problems with, it's just a lot of problems also with the digestive system and gastrointestinal and so on. But look at it, the brain cerebellum and the cerebellar hemisphere. This is, so if you were to remove this gene, you know, this certainly would be highly affected because these are the highest expressed in the so I thought, okay, what if we take the normal, like the human genome, and what happens when you remove that gene and so on. So that was sort of the rationale.
Liz?
Yes.
Quick question, what is TPM on the y-axis?
Transcript per million is the count.
Okay.
And that's how they do the sort of the unit for the gene expression. So I use the log of that because it's like in the millions and it's kind of big. So here's the log of the transcripts count per million. These are in alphabetical order the tissues, and this is removing the autism genes in the score 1. And these are, and so I'm putting there, I'm coloring in red those genes. And the question that I asked was, so if I color those genes in the autism gene score, where do they manifest maximally? Okay, so that's the, you go down here, that's the brain cerebellar hemisphere and the brain cerebellum and the brain frontal cortex. These cells culture fibroblast. I'm not sure what this, I think this is when they culture the cell lines and they build, so that's where they also, and so on. So this is the rationale. I said, okay, first of all, let me see where they manifest. I started to plot all these different scores and did the same for the ataxias and for Parkinson, and kind of visualize where is it that they're maximally manifesting in the tissues. And so if I were to remove them all together and you know, plot again the whole thing, what happens? So this is another, yes.
Sorry, I'm not sure I understand what is score 1. What are the scores?
Okay, so in the paper, let me go in the paper, in the appendix or supplementary material. Okay, let me just break out of this and find it. We can actually go back. I'm gonna just go to this site. There you go. Let me see. It's a whole set of criteria that they use. I want to go to it, but it won't let. Oh, maybe if I do this, hold on a second. Yes. Okay. All right. And so this is the SFARI gene module site, and as you see, it's being updated constantly. So let me navigate gene scoring. Okay. So, okay. they assign in the databases reflecting the strength of the evidence, linking it to development of autism. So they go through the literature. So these are these different syndromic, 1, 2, 3, and so on. So these are the number of genes. And so if you go to the category of syndromic, you see the number of reports in the literature. This is the genetic category. This is the chromosome where it happens. This is the name of the gene and the acronym, and so on. You go to category 1, and so you see the reports are larger, so higher evidence in the literature, and so that's the criteria they use.
Okay, so the most evidence would be 1, right?
Yeah, the most evidence is 1, then 2, 3, and then syndromic is a different category altogether because it's like very complex, it's interacting a lot of genes and so on.
Okay.
Can I ask a question?
Yes.
Most of these genes associated with autism, are they, although I know in reality there's not a clear distinction, are they recessive, dominant? And
all of that, it's a little bit of everything, and it's very complex. Like it's not a one-to-one map.
Yeah, I'm asking because does this whole thing imply that all over the healthy population, these genes, these mutations are present? So it means that either everyone is pretty much on the spectrum.
Right. Yeah, pretty much. And it's the combination and the environmental exposures and everything combined that gives rise to this. So it's not like a purely genetic disorder or anything like that of genetic origins, but it is, it has a high genetic component when you look at siblings and familial ties and so on.
So if it's not purely genetic,
yeah,
maybe it can be prevented. I don't know.
No, I don't think so. It's like preventing being human, you know, it's so inherently, so just you get more of one thing than the other. And the idea of this paper is actually not to say, is not to go and find the causes of autism, which is what everybody's trying to do. I don't think that trying to find the causes of autism is a tractable problem. I think it's totally intractable. I wouldn't go that route. What I want to do instead is go to what exists already that builds supports and accommodations for a person that has a neurological condition. So in the ataxias, for example, this is the case. And so by linking a subtype of autism that primarily has a phenotype that resembles a lot of the ataxias. Yeah, because clearly you cannot go and test everybody genetically in the population. That's not feasible. But if you get a random draw of the population and you see that phenotypically that person resembles the ataxia syndromes, then you can go to the Ataxia Society, which has a society, and see what kinds of accommodations do you have for people who have frequent falls, who cannot eat because they have tremor, who can, you know, and so on. These accommodations exist in the neurological world. So I'm trying to bring to autism these kinds of ideas, like here, you can help the person, instead of saying we have to modify their behaviors because they look socially odd. What you can do is drop that model altogether, which is useless and it's just hurtful. And what you can do instead is go to what already exists in medicine and see. And so what I'm trying to show is evidence that the overlap is so big that in fact, doing this gross random deletion of these genes, you do get neurological conditions. You know, at the beginning, this was a hypothesis, but we'll see what I got. I'll show you what I got, which then it's like, oh my God, indeed, these are neurological conditions, not like bad behaviors like they're saying.
It's like a guide from the genome to the tissue and to specified treatment according to what tissue and what type.
right?
brain.
Because now we can say, okay, what are the tissues that are most affected, right?
Yeah.
Okay, is the heart. Okay, this person has cardiac issues. Let's go and look at the cardiac muscle, which has specific enzymes and specific genes, and and then we develop a targeted treatment for that, right? Oh no, this person has a lot of cerebellar tissue damage. Well, we know that cerebellum is implicated in motor coordination, motor learning, motor adaptation. So we can create accommodations for this person and go further and investigate the cerebellum and the cerebellar genes and see we come up with a targeted treatment for that person that has cerebellar tissue damage because of gene expression. I mean, this is things that already exist. I'm not reinventing the wheel. All I did was repurpose what's already on the web. But so let me continue. So this is like, for example, the SCORE1 genes, and then you go, oh, these are all the genes in the SCORE1. Oh, choose. No. Yeah. Okay. So these are all these genes, right? And so this is another representation in sort of color map form. These are all the genes, and I'm plotting the log TPM here. So higher is yellow. And to visualize which genes, are higher in which tissue. So for example, here, again, the brain cerebellar hemisphere, quite yellow, right? So and that's pretty fundamental for, and this gene, actually, this gene over here is impacting everything pretty much. So you can get a sense of what these genes actually are doing in terms of these 54 tissues. This is an example of the Fragile X genes. And actually I'm pulling here the syndromic genes in, I don't remember what color, I should have labeled the legend, but these are all the Fragile X genes that the SFARI has for the different scorings, because they did also for Fragile X, since it has high penetrance in autism, meaning that a lot of the Fragile X, people with Fragile X also get a diagnosis of autism. So different. So these are all different genes of different scores. I don't remember the colors. I say green is 1, red is 2, and da da da. And so these are affecting, again, the brain cerebellar hemisphere, the brain prefrontal cortex, and so on. I mean, not affecting, I should say, maximally expressed in those tissues. So the hypothesis is what happens when you remove them, right? And so here is sort of a stem plot of each one of these different tissues. So now I can go tissue by tissue and plot sort of the gene expression across the different genes. So these are all the genes of SCORE1 and their expression in log TPM in the brain frontal cortex. These are the SCORE2, the SCORE3, the syndromic, and so this gives me a sense of kind of their expression. And this is after the normalization that I just said. So I put everybody on the same 0 to 1 scale. So this is like different, like basal ganglia, heart ventricle, just examples, there are 54 tissues, remember? And so then these are their frequency histograms. And then I realized, okay, so these are pretty much exponential here after the normalization. So I'm gonna actually, after I remove them, I see, so I removed the genes, for example, the SCORE ones, right, for the basal ganglia, and I see these. And so then I realized, okay, so this is, can be well fit by an exponential distribution. And so I'm gonna do maximum likelihood estimation and estimate the lambda parameter of the exponential distribution given this gene expression sequence. So the question that we ask is, given the known neurological phenotype of the known disorders like Parkinson or ataxia, X chromosome, Fragile X, and mitochondria, is there convergence between the most affected tissues when I remove those? that is according to stochastic change, right? So I take a stochastic change and the roadmap to machine learning driven detection classification. So eventually I want to have a neural, I want to have a network learn this classification, but we're not there yet. That's what Theo is going to help me do. Theo, no pressure.
Yeah, this part. Yeah.
Yeah. So
I can
anyways,
start.
yeah, we're gonna work on this. So count the values conceived as a random series of numbers and use maximum likelihood estimation to model the numbers as generated by an exponential distribution, where X represents the normalized counts using this equation 1 in the paper, and the value and Y is the value from the exponential distribution. And so we're gonna estimate the likelihood of lambda. So what the lambda parameter is, which gives you then the exponential curve. So the likelihood of lambda given the whole gene expression, X1, X2, Xn, for all these 54 tissues, right? Remember, each tissue has 56,000 something thousand genes, minus the ones we're gonna be taking, but that's like a random, I'm treating it as a random draw, yeah, just essentially. In the paper, I described how to get the lambda, just it's textbook kind of derivation, you know, you just using the log, the logarithm rules, and so you end up with lambda n over the sum of all these values. So given the sequence, you can get lambda. Given the series of genes, transcript per million, counts per tissue, we can obtain a characterization of the gene expression and visualize it using our methods. So you can then, because the exponential distribution is part of, it's a special case of the gamma distribution estimation, we actually can plot this on the gamma moments space and visualize it. So that's what I do also in addition to the lambda. And we can use something like thermal resistance to quantify the departure from having all the genes, you know, when you remove some of the genes. So actually, this is critical to put a number on these things and visualize it and quantify it. And so now, yeah.
This MLE thing, we can do it even without assuming a distribution.
Right.
I mean, just in case it's a weird distribution.
Yeah, a weird, yeah. And so even if we don't have a theoretical distribution, we can also estimate some likelihood. And we can even do a likelihood ratio and without need for a distribution. But for the time being, I'm using this gamma parameter space to visualize it. And actually, you can do this with all, it's like a plug and play thing. So I'm gonna now I've been looking at the schizophrenia genes and see how they compare. What is the difference? Because here's the thing, you know, Parkinson is like manifests in the old ages, right? So it's a neurodegenerative disorder. Now, in autistic adults, you start seeing that after 40, you know, by 40, you start seeing this because it's an accelerated. And you do see some people at 40 that have, and even in the late 30s, that begin to manifest Parkinson. But it's like over there in the old age. Then schizophrenia by 21, around 21 is when you manifest it. And then autism is like neurodevelopmental. So you see this timeline and this kind of asynchronous way of manifesting these neurological disorders. So that's what I'm trying to do, characterize also the timeline based on the genes. So here's what I do. I remove the genes. So this is, for example, all the autism genes from SFARI. These are all the scores. So the 916. And it has some it has some manifestation, some sequence of the tissues with all the 54 tissues with some gene expression after the removal. And what I do is I break it down, and this part I forgot to include in the PowerPoint, but you can see it in the paper. I break it down by taking a median rank the whole thing. I sort it and plot it, and then this is the exponential function for the black curve is for the normal, so all the genes, and the red is the shift that you get from removing it. But what I do is I median rank the delta lambda. So I for each one of the tissues I subtract this the outcome lambda from removing minus the outcome lambda from not removing, from having the entire genome. Okay? And then I sort it, and here's so the maximal, so it's sorted here, and it's the maximal difference here, the delta lambda, is in the brain putamen basal ganglia, then followed by the substantia nigra, brain amygdala, and so on. So this is the first rank, median rank of the 54 tissues in that order. And then I stop here. And so the pancreas is allowed, but there is the rest of the 54 tissues is just a smaller change. So they would be over here. So I'm going just by the maximally different, and this is how. Then I also plotted it on the gamma parameter space, estimating the 4 moments. Again, like we do in the other work that we do, median, mean, sigma, skewness, and then the kurtosis is proportional to the size of the marker. And so in open circles is just the full genome, and then this is the log TPM that results from the color circles is the shifted open gene removal. So this is a way to visualize this major, and this is the air mover's distance. So again, and organized by their movement, and you get a little bit of shuffling, but again, you do have pretty much basal ganglia, substantia nigra, brain amygdala, and so what you see is like these main guys here in the highest affected. What are they? They are the tissues in the brain and subcortical regions that are responsible for all the motor, learning, adaptation, coordination, development. So that's what you have in autism, right? By the criteria that sidelines all the motor criteria, right? These genes, remember, the scoring of these genes was done by that clinical criteria that specifically and deliberately says, no, there is nothing to do with motor, sensory, or anything neurological. This is just bad behavior. Here you have it. This is all the motor stuff, and the sensory motor, the neurological things. So now that we go and do the ataxia, Fragile X, these things, remove, all combined. Again, memory, emotion, and then substantia nigra. Here you have a lot of hard stuff as well, and musculoskeletal, but these are also part of that neurology taxonomy that I'm proposing, right? So I couldn't have made this up myself. I mean, this is stochastic. These are the ataxias, right? And again, aside from this whole blood thing, which is the highest, you again have all these regions here that are pretty much neurological. We knew these in the ataxias because that's what they are, but Fragile X, Parkinson, again, you have not surprisingly, because these are the neurological conditions that you have that are known. So amygdala, involved in emotion, substantia nigra, motor, brain hippocampus, memory, and so on. Mitochondria is different. So mitochondria has more to do with all these other all these other issues. It does have some memory and you know, emotion and some motor, but they are in the, I mean, this is the first median rank group, right? But there's over here, it's more like heart. So that's interesting. So this tells you also that within, this is another subtype of autism. It should be considered a different subtype because CUC, it's more of a heart related. Then I went on and looked at the overlapping genes, autism and ataxia, and I divided autosomal dominant, recessive, and then the X, autism and the X chromosome. So these are their expressions in TPM, and these are the genes that I came up with. And then, I mean, I didn't come up with the whole algorithm. The whole interesting thing is the supplementary material. If you go to supplementary material, I took the time to actually go to OMIM and take all of the information that I could about the phenotype and about the literature on all these genes. Each one of them is documented, tabulated, and the violin plots and everything, I'll show you just an example. And again, this is autism and Parkinson. There's one gene that is overlapping in both, and it's in the SFARI database and in the literature. And this is the log TPM. And then again, you know, amygdala, cingulate cortex, basal ganglia, brain, very neurological. And then towards the end, also heart and pancreas. This is the non-ataxia overlap. So I take just the SFARI genes without the ataxias. And again, it's not just that it has the ataxia genes, which has these neurological issues. There are other genes there that are contributing to the neurological issues that we know in the sensory motor, somatic sensory motor systems. And the same for autism and Fragile X. When we get the genes that are overlapping and we remove them, you still have the amygdala and the motor tissues and so on. This is just another way of showing everything together, but you can see, you know, the tissues that are maximally affected by all these things together, you know. So pretty much that's it, you know, that's the paper. So removing the SFARI autism genes alters gene expression maximally in tissues related to motor control, motor learning, adaptation, and coordination, followed by tissues related to emotional memory. There is overlap between maximally affected tissues in well-known neurological disorders like ataxias and Parkinson's disease, and those maximally affected by removing the SFARI autism genes. So motor, emotion, and memory issues can all be neurologically addressed along with accommodations we have from these other fields to help patients cope and flourish in life. And so I'm asking, is it time to change the autism model? Right? So that's the paper. Now, this is what is in the actual supplementary material. You will see I compiled all those genes that are overlapping. This is one example, CACN1C. And then if you go to it, so this is the cellular level, or is being scored here again, similarly as in the literature, the likelihood of this being affecting which part of the cell. So the higher here, the higher the gene is not producing some enzyme or protein involved in that part. And again, these are the tissue, the main tissues that are affected for the brain. This is just to show sort of for each gene, sort of, and again, so these are the genes that are, and you can see the most ones deal with the cerebellum and so on. So this you can go through that. So that's in the supplementary material of the paper.
The heart, how it's affected by genes, and whether there is a correlation between heart diseases and these neurological disorders.
Yeah, there is. So, I mean, we've seen that, how do I stop share?
I mean, I know that heart diseases are more common in males, and also autism is more prevalent in boys. So maybe there's a connection there. I don't know.
Well, there's it's bound to have, because those genes are present in the whole population with this diagnosis, and Mona is seeing the dysregulated heart in the kids. I mean, not all of them have it, but and so on. So that's sort of the message is, why treat this as a behavioral disorder when here is unambiguous, undeniable evidence that this is a neurological disorder, and it should be treated as such, and we should be learning from other fields that already build these accommodations for these people, and just have even targeted treatments based on the genome. In the ataxias, you have several of these targeted treatments. So anyway, so that's sort of my message. Is it clear? Yeah. Okay. Any questions? So Theo, we're gonna train networks to do this classification.
Yeah, and for the Fragile X.
Yeah, for the Fragile X, for the ataxias, and I'm now compiling all the schizophrenia genes. So there are like hundreds of them now that we could also do this with and see what is affected in the schizophrenia case, because it's bound to be different, because the timeline of the schizophrenia disease disorder is different than the I should be careful and say disorder, because disease turns out has a medical meaning that they know to some degree the origin, and we don't know the origin of these things, but disorder in this case. So what are the tissues maximally affected in schizophrenia? That's now a tractable question. And based on those tissues, how can we then look at it using other disciplines other than psychiatry? Because psychiatry, I mean, what is it doing to help the person? That's my point. What any of these psychological or psychiatric are doing to actually help the person live day-to-day activities of daily living?
Yeah, they drug them, basically.
I mean, isn't that at the end of the day, like, you need to prepare a cup of coffee and a sandwich and eat it? I mean, isn't that, or do you need to depend on another human being who does that for you, and is angry because it's not paid the amount of money he wants to be paid, and he humiliates you and abuses you, and so on and so on? I don't want to be in that situation, because we're all going there, by the way. We're all going in that direction, and at some point we're going to depend on another human being. So I want rather to have accommodations that allow me to have agency and independent living.
Is that
And that's
not,
the point.
Is that not happening now? Like,
No.
Training, because in Israel, at least, I spend a lot of time in
Israel. Is different.
Yeah.
Planet with very advanced everything, my dear. I've been there.
Yeah.
Pretty impressive. I'm like, why
I
can't
didn't
we do
know,
things like that?
I didn't know we were advanced in anything. I really, actually.
Because you live in it, and you don't have, until you learn, you live in it until you take it for granted. When you leave that system and you go to a system, I mean,
yeah.
The US has superior things in other things, but in this part, it sucks. It's like the person really suffers. And it's because this whole medical insurance system in Europe is also superior, not just Israel. Europe has a lot of different things at the school system that is way superior, but particularly I found.
What country in Europe?
Depends, right? Depends. But I mean, the Nordic countries are number one in education. The US is like number 10 or something.
Right.
Well,
I mean, it's,
Anna, to answer your question, one of the groups that I work with is actually an organization that manages group homes and day programs for disabled adults, wres. And I'm close with the executive director there. And her biggest problem is that they can only afford to pay their people $15 an hour. And, you know, if you're familiar at all with the US, that's what everybody's fighting for, $15 an hour minimum wage. And if they raise the minimum wage, why, her biggest concern is why would somebody come there where they run the risk of getting hit, having to take care of an adult's needs, everything from changing diapers to washing them and feeding them, when they could go work at Walmart for $15 an hour or,
right.
or anything like
so
that.
there are no people. So this is a problem now with COVID, because those people are in high demand, and they're highly, it's highly contagious, because you're dealing with human excrement, you know, feces and food,
Right.
and saliva, and you know, this is not an easy thing to handle, because the nervous system doesn't have autonomy. You have serious damage in the smooth muscle, that so you have incontinency, and you have, it's not pretty. It's not life like we know it at this young age. It's not pretty. I've been there. I know what I'm talking about. And so there is no system in place that help these people live with any kind of dignity. You have somebody who's angry because it gets minimum wage with a lot of risk. And now with COVID, they're going to a different job. And so why do you think the highest infection and death rate occurs at the homes?
Yeah, because they're living, you know, in the group homes. Now, wres, luckily, and to their credit, have had no deaths and only one infection in all of their residents. Their staff, they've had some more infections, but they literally locked down before Governor Murphy instituted the quarantine, and they kept all their residents at home. But their homes are 2-3 bedroom homes, and they have 5-6 residents. You are expected to have a roommate as an adult. These people, and not just sharing a house roommate, I mean literally like a college dorm style, 2 beds in a room, 2 people in a room kind of roommate. And that's all they can afford, you know, in a lot of cases.
I think a lot of this is also happening in Israel, and there's no money for anything. What I meant is that I thought that there is a bit of a greater consensus that it is a neurological.
Yeah, that's my point.
And I'm surprised that you're saying that there isn't, because that's what I'm used to in Israel. You have no money, but you know that what you should be doing is teaching like life skills and trying.
It's a problem of society, because like in many societies, like for example, when they still label as crazy someone that is schizophrenic, or they like use the, I mean that happens everywhere. Like they use the term autistic as an insult.
It happens everywhere, but to a certain extent. So in Israel, and I don't know all the details because I've been there only a few times and for a week or two at a time, so I don't know the in and outs. But what I know is in the academic world, the people that I work with and collaborate with have a very different frame of mind than here. They don't see this fight that I have to have in the United States, where my papers are it's never a smooth ride for me. Just because I'm saying, look at the neurology, look at the underlying biology, look at these, forget about this causal thing, which is not solving any problem really. You know, it's help the person that is affected by this condition.
Yeah, that's
what I see in Israel. Right. My collaborators, that's what they have in mind. Let's life skills and help the person, you know, this and that. My collaborators in Tel Aviv University in occupational physical therapy, that's their philosophy. So when I went there, I fit it like a glove. I mean, it was like a glove to the hand. It was just, yeah, what are you talking about? This is the norm. Yes, not in the US. In the US, I have to fight the system just to get this message across that, hey, there are alternative ways to look at autism. Look at Europe, look at Israel, look at other countries, and you'll see that other accommodations exist. In the 70s, this was how it started. But somehow it died out because behaviorism took over, built a business. Not that I say there are books out there explaining the whole business model of autism very nicely. And then you understand it. It's a revenue stream generator. And I can see why it's this way. And I can see why it's the people who are on top of this chain, the food chain, don't want to change it and give scientists like myself a hard time when I'm trying to point out. So I don't think this paper is in Nature Scientific Reports in the review now for a month. I don't think it's gonna be published there. So I just uploaded to the BioRxvi so that people can read it and so on. And then I'll publish it in some open access like Frontiers or one of those, NDPI, whatever. That's my strategy.
And I think that part of the problem is that autism is diagnosed when the parents still have full control.
Right.
And they want their child, they have dreams for their child, and they want their child to behave a certain way and fit in and look like everyone else.
Yeah.
And when you have everyone saying this is the only way to do it,
that's the way it is. Yeah.
they don't know what to believe.
Yeah. So yeah,
they
totally. We're dealing with it in our center of excellence. We see the disparity in the generational parental disparity in criteria, in what they want for the kids. It's very poignant because you're in a situation where this is what you're offered. You're not offering any other alternative, and the science doesn't have a real voice in it. And so, that's what it is. You get, now the younger generation is not taking it. They're just like, no, wait a minute. There are 2 decades of neuroscience. I know this is not the only approach. It was viable in the 70s, but it's been 40 years. Let's just give it a break. So, okay, so stop recording, Richa. I think we covered all the grounds here, and then let's